Okay, the screen recording has started. So now you can see here we have these many proteins like Arbus, Ebulin, okay, PMRIPM, PMRIP2, Ricin, SNAV1, SNAV5, Viscamin, Volkensin, RCA, MCL1, Ebulin1, Abrin A, HMRIP, SGSL. Okay, and you can see this all these protein sequences, the score is around 93, 92. Okay, so all these proteins are from different plants with same function. Okay, the sequence, these are the proteins from different plants but they have the same function. So you can see the conservation of the sequence. So it's around more than 90. Okay, except for this some volcanism. So if you look at the average score is 92, and you can see here the coloring pattern also. So if it is bad, if it is completely bad, it will show gray, and if it is, then we have blue, then we have green, light green, yellow, followed by light pink and the pink. Okay, so pink means very good. So now if you look at the sequence, you can see the sequence here. First, initial regions, so there is a lot of gaps. Okay, so there are a lot of gaps, and within this also you can see there are some regions which are having same residues. Like here you can see all the residues are tyrosine except for this SGSL. So likewise, when we look at this protein, you can see here these regions, they have a lot of regions which are highly conserved. You can see this one. So here you can see all the amino acids are L. So it is represented with the star. Okay, so star means conserved. Okay, and you can see here this YF. Okay, can you see all the F's? Then we have YFF. So what is Y? Tyrosine. And what is F? F is phenylalanine. Tyrosine and phenylalanine, both of them are aromatic amino acids. It means their physiochemical properties are same. So that is the reason it is represented by colon. Okay, their physiochemical properties are same, so it is represented with the symbol colon. And you can see here we have all F's, and in between there is only one H. So this, all of this, they have phenylalanine is aromatic, we all know, and histidine, it is not aromatic but it has pi electrons. Okay, like it has the ring structure, and you can see it is represented with a dot. Okay, and whenever it is blank, it means that region is not conserved. Okay, but still you can see in this region, so there are very less gaps, and this is the functionally important region. So that is the reason I have marked here. Okay, so I want to uh, see the differences in this region, actually this Y and V. Okay, so you can see some amino acids, some proteins, they don't have this Y and V, they have a different amino acid. Like here you can see it as VA. Okay, so it means the proteins which has Y and V, they are functional, which is having something else, they are non-functional here. Okay, that is the conclusion here. Okay, so generally what we'll do, whenever we give multiple sequences and ask the tool to align, so it will align in this way, and it will show with the gaps or the sequences, and it will show, this is the thing we need to look at actually. It is shown as star or semicolon or dot. So here I am taking many sequences, like we have around about 10 plus sequences. So that is the reason the conservation will be very less. Suppose if you are comparing only human insulin and pig insulin, then you will get lot of stars and lot of columns. Okay, ma, and very few empty spaces. Okay, and you can see some other region also, like here we have all G and Y, and here you can see all the amino acids are E, and it is given as star. And here we have R and K, arginine and lysine. R is for arginine, K is for lysine. both are functionally same, so that is represented by colon. Okay, so like this we will develop a multiple sequence alignment using any tool. Okay, so a lot of tools are available. In the further classes we'll discuss them. So, and then we'll look for the functionally important residues. Okay, whether they are conserved or not. Okay, or else if we don't know anything, then we'll compare and we'll find out which region is conserved, which regions are not conserved, and based on that we will draw some conclusions. Okay, if the regions are conserved, it means these are functionally important regions. If the regions are not conserved, they are not, they are functionally not important. Like that we classify the different regions. Okay, so you can see another residue here, W. Okay, so this is also conserved. And the ending sequence, you can see there are a lot of gaps. So usually at the N terminal and C terminal we will see a lot of gaps, and in between there will be the functionally important residues. So those regions will be mostly conserved. Okay, hope you understood this one. So then we'll continue with the secondary structure prediction part. So first tool, we are going to use the same sequences that we used in the last session. So the first tool we are going to look at is PSI Pred. So whenever you are writing in your thesis or notes or you are writing exam, write in the same manner. Capital P SI is small and capital P PRED. You should write, follow the same pattern, PSI Pred. Okay. There should be two capital P's. So just click on this tool. So this it has lot of tools within this, but we are going to use only PSI Pred. You can see here also we have signal pep sequence prediction. Okay, so here also we have domain prediction, function prediction. Okay, but whatever we have discussed yesterday, those are the best tools. Okay, so now let's take the sequence. So let's take this sequence 1. Let's paste it here. Then you can give an email ID. Okay, and then click on submit, or you can give the job name also. Okay, if you are doing submitting multiple jobs, so always give the job name and give your email ID also, then click on submit. So it is saying job name is required, so let's give the job name here. Okay, submit. Okay, it want email address also. It is actually here it is written optional, but it is mandatory. Okay, then click on submit. Okay. So meanwhile, I just want to tell you, those who have not registered till now, try to finish the registration process by Monday, no? And you can also, if any of your friends are interested, still they can join. Okay, till Monday they can join. After Monday, I won't recommend. Okay, so if any of your friends are interested after hearing the feedback from you guys, so they can still join till Monday. Okay. Why this is not working? So these tools basically, the servers most of the times we have this issue. Okay, so the server may work or may not work. It will not work today, tomorrow it may work. Okay, so this usually happens. So mm, so this will produce two types of images actually. So one image is this way. Okay, so this is one image it usually produces. You can see here this yellow color indicates it is a strand. It means it is a beta sheet, and this pink color indicates it is a helix. Okay, and you can see the remaining all it is in gray color. It is a coil. Okay, so if it is a dark gray, it is a transmembrane helix or it is interacting with the membrane. And you can see then all these things are gray in color. It means it is a coil. It is just a thread like structure. Okay, and you can also see the color variations here. So if it is blue color, okay, and white, it is disordered, and it is white and it is with green border, it is protein binding. Okay, so we usually think if a protein does not have structure, it is non-functional, it does not have any significance, but that is not the true part. Okay, so even the protein does not have any structure, it has still has a role, so that can bind to some other proteins also. That is the reason here it is showing disordered and protein binding with the green colored rectangles here. Okay, green colored squares or rectangles. Okay. So what the conclusion or the take home message here is that the region which does not have any structure, still it may have a functionally important role. Okay, so nowadays people are doing lot of research on this kind of proteins also. So we call them as intrinsically disordered proteins. So the protein does not have any structure, but they are functionally very important. So the lot of research is going on. So till a few years before, people used to think if a protein want to do some work, it want a structure, but that is not true now. Even protein without any proper structure also has a role in doing some work. Okay, so this is one map. Okay, sequence based map this way, and we have one more graph also you can see here. So this is the sequence, and you can see this is the beta sheet, and this is the pink one is the alpha helix. Okay, and you can see the confidence bars here. Okay, this is the bar diagram, confidence for each residue, and it is showing it is a prediction, it is coil or beta sheet or helix. Okay, so C, E, H, those are the codes for the prediction. Okay, C means coil, E means strand, H means helix. Okay, and you can see the amino acid here. MA is, this is the amino acid sequence. So this is another map showing the confidence course also. Okay, so no need to worry, you can see the like graphing pattern. So if the graph is bar size is less, it means the confidence is very less, and if the bar size is very big, then it is the confidence is very high. Okay, and so this is called as three assignment cartoon. So like the line cart is the cartoon representation. So strands will be represented, beta sheets will be represented by yellow color, and alpha helices will be represented by the pink color. Okay, and this prediction is three-state prediction. It means C, E, or H, and this is AS is the sequence. So this is how the result of PSI spread appears. and that we read it in this way. Okay, and now you can see here this region, uh, you can see from here 21 to, so not 21, this is, so this is 151, it means 170, 171, 171 to 202, 171 to 202. What is the length? 30, right? 31. So this entire stretch is 30 amino acids long. It means, so, and it is a helix. So it means whatever the protein we have taken, maybe this protein is a transmembrane protein. Okay. So this protein may be a transmembrane protein. So this way we can draw some conclusions also. Okay. And now we have to club this result with the TM-HMM helix prediction tool, and we have to compare it is showing in the same region or not. So what we'll do, we'll club the result from these two tools and we will conclude that the chance of the region 171 to 202 is has a high chance of prediction to be a transmembrane helix. So like that we are going to draw our conclusions. Okay, so that TM-HMM helix tool analysis and this secondary structure analysis, both the results we club and we write our statements in the discussion part. Okay, so this is like two different things, that is primary structure analysis, this is secondary structure analysis. So both of them were coinciding with the result. So it shows that whatever we are doing or whatever the conclusions we are drawing has some valid points to, okay, to conclude or to confirm. Okay, is it clear? Yes, are you all active? Okay. Okay, ma'am. So I hope you understood this one, and unfortunately this tool is not working today. So now let's try with the same sequence. We'll take the same sequence. We'll try with JPRED. See here, based on this sequence, what does this sequence has signal peptide? Can anyone answer this question? Does this sequence has signal peptide? Yes or no? If yes, why yes? If no, why no? At least tell yes or no.
No, sir.
No. Okay. Okay, you people are right, because if the signal peptide is present here in the starting region, as we discussed, it should have a lot of leucines. Here it does not have leucine, and this we are doing secondary structure analysis. And what I told in today's session also, the signal peptide always it will be a helix, and you can see this starting region we don't have any helix, only this small region is having the helix, and here we don't have any leucine residues also. So if it has a signal peptide, from the starting region only it will be helix, like from third residue mostly it will be a helix. Okay, that is also one standard point to conclude that this sequence or this example does not has a signal peptide. Okay, so now let's look at the JPRED. So this is something else we have taken to understand the result part. Okay, so now we are going to do analysis for this sequence one. Okay, let's copy this and JPRED. So here we need to paste the sequence, remove the existing one, paste your sequence and click on make prediction. And similarly, in the interest of time, we will open the other tool, Stride also. Stride. Okay, in this tool we can give the PDB ID as well as we can give the sequence also. Okay, side secondary pressure prediction server. Okay, so this is the home page for Stride, and you can see this is web interface. Okay, so here it is showing directly only for the 3D. Let's see extended input options. Okay, here also we don't have. And you can also install in your system, but you need to know the Linux commands. Okay, how to use Linux and the small basic shell scripting you need to know, then you can use this one. So here in the web interface, previously we used to have for the sequence also, but here we have only for the PDB structure. So JPRED is under Q. Okay, let me check whether we have any other server for Stride. So whatever I am doing the searching part, just try to remember I am using the word symbol plus plus most of the times. So plus is like it will combine the two inputs and it will give the output. Okay. So let's try to go from this research article. So whenever you can't find, you are not able to find the link, then you can open the research article associated with that particular tool and you can scroll down. Mostly in the abstract you will get the sequence, or somewhere in between you will get the sequence. Okay, so just click on the sequence and it will take you to the web server. So again we have landed to the same site. So it means they have removed this sequence submission one page. So here we need to upload only the PDB data. So for this we have to take the PDB structure from protein data bank. So do you all know what is a protein data bank? No? Okay. Okay, ma. In that case, so how many of you are in 5th sem? Okay, 4. So ma, till this semester, do you have any course like basics in bioinformatics or any course related to bioinformatics? No. Okay.
So we have it in the upcoming semester.
Okay, you have it in next semester. Okay, fine. Okay. So no worries. So anyway, you are able to understand the concepts and all. And as I'm taking it from the basic part, no worries. So what we'll do, I will include a. See, it's not like I'm teaching the course or something. We have to run and finish the things. So anyway, we will finish it in time. But at the same time, we need to know some basic part also. So what we'll do. We need to know a little information about databases. Okay, ma'am. So whatever the tools we are using in the previous class and this class, they are the servers. Okay, so they work on different algorithms and they will analyze our input or query and they will give the output. Okay, so like whatever the sequence you have used today for doing the assignments, so all those sequences I have taken from the databases. So we need to have a little knowledge about databases. So what is meant by a database, and what are the different databases we have? That basic info we need to know. Okay, that anyway I will introduce you that part in the next session. Okay, uh, not in we are not going to read those parts in detail, just a basic info and how to use them and how to retrieve the data. Okay, how to retrieve a protein sequence, or how to download a pdb from protein data bank. This basic part we will try to cover. Okay, and anyway that is also required for the further sessions also. So you can see we have the result, view result summary and svg, or you can download the result as pdf also. I think it is taking time. So this tried we will do it. Yeah, we'll do it today. Anyway, we have some time. Okay, we'll download the PDF. I think that will be better. Okay, you can see here we have the amino acids here. So we have given one query. So this is our query, and these are the different like reference sequences it has taken. Okay, to develop the secondary structure. Okay, this is all not required. So this is like this is a multiple sequence alignment actually. And you can see here this is the structure. So this is the structure, secondary structure actually it is showing at the end pages, at the end of the page. Wait, just let us go to the last page. Okay, now so you can see here we have a few. So this is not actually required for us. Okay, this is around 210. Oh, excuse me. So from here we are starting. So you can see again we have L here. So it is using a different set of tools like JNet, JHMM, JPSSM. Okay, different set of tools, and you can see this regions it is identifying as the helix. So this is helix and this is also helix. And if you see the second page, you can see these are the beta plated sheets, this yellow colored arrows. So it is using three different algorithms. One is JNet, JHMM and JPSSM. Okay, and you can see here we have an beta sheet and followed by an helix. So we can look at the different outputs here, or you can this you SVG is the actual one that gives only the required in data map. Okay, so when we are looking in PDF, it is giving the all background work also what it has done. Okay, so in the background it is doing multiple sequence alignment with all the available reference proteins. You can see here it is uni reference 90. What does it is saying? Uni means it is the database called UniProt, and reference it is taking only reference sequences, and 90 which are 90% similar. Okay, it is taking the tool in the backend. It is taking our query and it is comparing with all the reference sequences which are present in the UniProt database and which is having at least 90% similarity, and then based on this it is giving the output of the secondary structure. Okay, yeah, do you have any doubts in this? Then simply we can download this the zip file, and you can look at the SVG image. Okay, here it is. The SVG is not supporting it, or it will take some time to load. Okay, so let it load, and meantime I will tell you. So for this stride we need to give a PDB data as an input, or PDB as an input. Okay, PDB means the 3D structure. Okay, ma'am. So the theory part we are going to do in the next class, but let me give you a demo. Like we have a database called PDB, RCSB PDB, Protein Data Bank. So this is a repository for all the 3D structure of the proteins. Okay, so here it is a repository for all the 3D structure of the proteins. So let's take insulin. And you can see here we have around 13,078 structures. Okay, so now as we are interested only just to understand how to download, so just we need to click on this download file. So that will download the file in this .zip format. Okay, so we don't want in this .zip format, or with this is a file you can what we can unzip and look. So right now what we do, we just click on this one BOM, that is the PDB ID. Okay, so for every structure present in this database, it has previously it used to have four digit or four letter alphanumeric ID. Okay, we call it as alphanumeric. It means alphabets and numericals, A to Z and 1 to 9. This combination now ID. So this is 1 BOM. Okay, four letter ID. Okay, four alphanumeric ID. So but now because of too many structures being deposited, it is being changed to 8 letter ID. Okay, ma'am. So it is given as a 8 letter ID now, identifier or ID. So still we can use this 4 letter one only. So now you can see here we have option called download files. So just click on the PDB /MMC format. Okay, or you can click on this legacy PDB format. So this will download only the PDB format file for this structure 1BOM. So you can see here, so this file has been downloaded 1BOM.pdb. Okay, man, just open this in the folder. There are two options here, so I will show you both the options. So the first option is you can go to the Stride server, you can choose the file, choose file. Okay, we have three options here. Okay, so you can click on choose file, and this you all know very well. Like we have to select this file. Okay, this is one way, or the second way is you can type the PDB ID here 1B0M. Okay, so this is the second method. And third method, paste your PDB data. How you will paste? So this first two I know that even without guidance also you will be able to do, but the third one is a little tricky. So whatever the PDB you have downloaded, so this is the PDB. So give a right click. Okay, so give a right click on this and click on open with notepad. Okay, so you open this file using notepad. You can see the header. What is the header here? Insulin-like brain secretory peptide. Title is three-dimensional structure of bombixin 2, an insulin-related brain 2 secretory peptide of skill moth of Bombyx mori. So this title is given in 3 lines. Okay, ma. So it is showing 2, 3 here. Okay. So this is the molecular ID and what are the source. So this we'll try to understand this in next classes. So you can see here, if you scroll down, you can see the after remarks, you can see the sequence here. Okay, atom. So first atom is 1 nitrogen. So this is from the amino acid glycine. I hope you are able to understand. So this is the atom and this is the position 1. This is the atom nitrogen and this belongs to the amino acid glycine and this belongs to A chain. Okay, and this is 1. Okay, so this is the first amino acid. Here 1 indicates the first amino acid and these are the x coordinates. Okay, and this is y coordinate and z coordinate. 3D structure, right? So in 3D space the molecule has x coordinate, y coordinate and z coordinate. So all the coordinates, and then this is occupancy and this is the B factor. Okay, so all these things are present here. We just need to copy. You can copy the entire thing. Copy, simply Control A and copy. We have to open the PDB file in Notepad and copy everything and simply we need to paste here. Okay, so then we can click on compute or visual. So I'm just reloading the page. So we'll do all 3 ways. So here we have the stride. Oh my god. Okay, it got struck. Okay, I will try to close some tabs. Okay, close the program. Okay. Okay, finally, the Chrome has been closed, so I have rejoined the meeting here. And hope now the screen is visible. Okay, so we are using the Stride. Okay. So here you can choose the PDB file. Just click on choose file and select the PDB that we have downloaded and just click on visual. Okay, run Stride and produce visual output. Okay, so that will run the Stride and it will give the output. And you can see here this insulin usually it has three chains actually, chain A, chain B and chain C also. So C is, we call it as C peptide. So generally whenever we want to know what is the quantity of insulin produced in our body, we go for C peptide analysis. Okay, so that will tell what quantity of insulin is produced by our body. Okay, so I don't this server is throwing the error, or maybe the internet is slow. And you can see here it is showing alpha helix, extended configuration of beta sheet and isolated beta bridge. Isolated beta bridge, it means small beta sheet. Okay, when it is showing T, it is a turn, C is coil, and it shows 310 helix and pi helix also. So this is what we have discussed. The normal helix which forms the normal helix is called as 310 helix, and we have I have discussed about the pi helix also, right? So this tool, it gives information of all the, like, this gives more concrete information than any other tool. Okay, so unfortunately this is not working. You just give a try from your end. So take like in the yesterday's assignment. Okay, so in this there is one transmembrane sequence, right? Most of you have identified the transmembrane one. So that transmembrane sequence you uh, do the analysis, okay, using Stride as well as the JPRED as well as PSI PRED. Okay, so we'll see how it works. Okay, so anyway, in tomorrow class also we'll try to repeat the things because the servers are not working. That is not in our hands actually. Okay, or else let me show the slide. So Stride will give an output this way. So you can see Emma, so this will give an output this way actually. So chain A, you can see this is the turn and this is the extended configuration of beta sheet, and this red one is the alpha helix, and again this is the beta sheet, and these are the yellow color lines are the turns, and red color one are the helices. Okay, and green color one are the beta sheets. So this is how the stride gives the output. Okay, so unfortunately we couldn't see the output for both the tools today, PSI Pred as well as for stride. Okay, so we'll try to do it in next class or some other classes. Okay, so let me take attendance for today. So today is 25th, right? Okay, Soumya is
Yes, sir.
present, Paripurna is present. Okay, Sabha is present, Marvin is present, Zoeb is present, Seba is present, Nikhil is present, Suchitra is present. Suchitra, have you registered, ma? Yeah, Suchitra, are you there now?
Yes, sir.
You have joined in between. Yeah, have you registered?
No, sir.
No, ma. Okay, when you are planning, ma?
Sir, after Monday, I will complete the registration process.
Okay, okay, ma. No problem. So, Akshaya, are you present?
Yes, sir.
Okay.
Hello.
Yeah, okay, ma. Caroline, Bhumika is present.
Yes, sir. Present, sir.
Caroline is absent today. Yeah. Pravalika,
Present, sir.
okay. Srija,
Yes, sir.
Srija is present. Varshini,
Present, sir.
okay. Sindhuja, Manasvi,
Yes, sir.
yeah. Sindhuja is present.
Present, sir.
Fasiuddin,
Present, sir.
okay. Varun is present. Navin is present. Bharat,
Yes, sir.
okay. Sweta Varshini, Sweta Varshini, are you present, ma? I think she is absent. So, Aryavardhini, absent. Kiranmai, Kiranmai is also absent today. Mahesh, I think Mahesh is present. Sriya, Ramesh,
Present, sir.
Ramesh is present. Ritika,
Present, sir.
Sahan Shreya, okay. Kiranmai is absent, right? Aryavardhini, Sweta Varshini. So all three are absent. Okay. So I just want to tell you, those who are not done with the registration process, try to finish by Monday, ma. So if anyone else is planning to join, they can also join till Monday. Okay. So later on also they can join, but it's like it will be difficult for them to understand the topics. Okay, if the time is being killed. Okay. Okay, ma, thank you all. Let's sign up for today, and tomorrow also let's meet at the same time. So tomorrow is holiday for you guys.